central nervous system. But before that, we should know that what nervous system is. Who's going to help me? What is nervous system, or how would you classify nervous system? Anyone? Okay, mainly you should know that it consists of peripheral nervous system and central nervous system. Peripheral you have already done in your first year regarding microanatomy, that you did the nerves, the cross section and LS, and the ganglia, right? So today we are gonna discuss the CNS, in which you must know that brain and spinal cord are the major part, right? And among the brain, you've got the cerebral cortex. So in this picture, what striking thing you can notice? Yeah, sulcus. Which sulcus? Any main feature that separates the two? There are two cerebral hemispheres. So what is this?
Corpus callosum.
Corpus callosum is gonna join them. So what that fissure is?
Median fissure.
Okay, medial longitudinal fissure is there. You have got two cerebral hemisphere, and you see that it is being thrown into various folds. The elevated area and the grooves or depressed areas. So the elevated areas are the gyri, and the depressed or grooved area are the sulci, right? So gyri and sulci. So you can observe them in a whole pattern. And why they are there? Because the brain is now showing convolutions, and all this is due to or meant for increasing surface area, right? If these were not there, a plain surface could be there, we cannot have this much information which we right now have. Okay, so these foldings are now having so many brain tissue in this space, right? You get it? Okay, and what else we can observe regarding this? We can have another image. You can observe two cerebral hemisphere, and in this lateral view you can observe various lobes. You must have already learned about it, the basic divisions, that you have got the frontal lobe, the parietal, the temporal, the occipital, and inferiorly what you observe, you get the cerebellum, and if midbrain is there, then you observe the pons and medulla oblongata. These are the main parts, you would say. And this is the sagittal section to make you clear that what structures are on the upper surface, and you observe the corpus callosum and all the structures. But we are today concerned that what this cerebral cortex is made up of. When we talk about the cerebral cortex, we mean the gray matter. It means our cerebrum is having gray matter on its outer side, right? White matter on its inner side. So, can you help me telling in this what gray matter is? How would you define it? Yeah, collection of cell bodies. So, we have got collection of cell bodies in the ganglia as well. So, what we should say? Yeah, this is outside, inside, this is a term, right? So when we get this collection inside your CNS, right, we collectively call this as gray matter. So can't we say that those ganglia have got that gray matter but outside, okay? Still having the nerve cell bodies or neurons in it. So your gray matter is basically the seat of intelligence. Whenever we want to say a person who is dumb, we say कि जी इसका gray matter नहीं है. You understand? So all the functional activity is concerned with your gray matter. So basically, neurons lie there, and of course, initial segment of some unmyelinated, myelinated axons could be there. Along with the neurons, you get the supporting cell as well. But predominantly, you said neurons are there. White matter is meant for the all the axonal fibers predominantly. Few other supporting cell could be a part, right? Okay, this is the main thing. So let's move towards some other pictures which will come across and we'll discuss one by one. So You are looking into the picture of a neuron. It is just like that your cerebral cortex is having a place which we call it a seat of intelligence. So many engineers, so many doctors, so many scientists, they are sitting. So we should know their name. So among these, we categorize them as a pyramidal cells or pyramidal cells and non-pyramidal cells, right? Okay, so can we name them? The pyramidal cells and non-pyramidal cells. Okay, the pyramidal cells, as the name tells us, the neuron should be having the flask shape or triangular pyramidal-like cell body, right? And what parts are associated with the neuron? That it should be having the dendrites, right? These branching processes are the dendrites. Okay, in the center of the base, it has got a longer axon. So these are the receiving ends, and the axon should reach somewhere to either make synaptic connection with the other. So this is the look of the pyramidal cells. So we again divide it into three on the basis of height of these cell bodies, right? They could be small, they could be medium, they could be large. Usually the size being written is 10 to 50 micrometer. But it can be so large that it can attain a size of 100 micrometer. So sometimes you get the largest giant cell called as bed cells. BETZ, bed cells. They rest where? In the primary motor cortex of your frontal lobe, right? Okay, so that was about the pyramidal cells, which you are also looking in the screen as well. There is another category of the scientists, engineers, or doctors sitting, which are called as non-pyramidal. So let's hear the name. The horizontal cells, the granule cells or stellate cells, then comes the fusiform cells and the Martinotti. These are non-pyramidal neurons. Most of them could be interneurons or you observe them as a motor neuron. And we should know that these pyramidal cells are motor neurons. So the most frequent neurons in the body are motor neurons. So again, you can see this histological section that there is a cell body or soma or perikaryon having the nucleus, nucleolus, all the cell organelles. Then you see so many branching pattern called as dendrites, and there is an axon. Right? Is it clear? So now we will see some other pictures related to it. This is a repeat. Okay, that was a general concept that how you get information, they are ascending or descending through various pathways and various fibers. On this sectional view, you are observing this grayish color outer structure called as gray matter, right? And the central white structure is the white matter. So almost 70% of your cerebral cortex CNS is made up of white matter. Okay, so outer gray, inner white. But there are definitely buried again gray matter known as basal nuclei or basal ganglia. Okay, is it clear? And you are also looking to some fibers like corona radiata and then internal capsule, and you can observe later on when they have to pass through and through the medulla oblongata, its pyramid, they have to cross, and there are so many decussated tracks. So this is the concept that the track or fibers or funiculus, they are carrying the axons, and the commanding center is your gray matter. It's going to receive the sensory info, like as if you are looking into the picture, you are receiving something through your receptors of your eye. You are hearing through your ear apparatus. All the information are being taken through the sensory receptors as being afferent, being a part of your cranial nerve, right? And definitely if you move your body, the spinal nerve are also getting info, and all these are being reached towards your cerebral cortex in the gray matter. There they are appreciated, processed, then there is the integration, and then we have to do the action accordingly. Motor action, right? That if we are hearing, we can move our body. If somebody is asking us to take something to pick the book, to move, so and so. So all this action is being decided in your cerebral cortex, and these neurons which we discussed, they are making connection how. So let's see if there is a roof of your classroom. You see it's being lit. Can you focus them? This is very important. If you can switch the bulb, just you are receiving a sensory information. You are giving. They are suddenly, all of a sudden lit. Within no time, these are the neurons, right? And they have got circuits. They are interconnected. They are making synapses. This is how you are understanding the thing. Right? So sensory information is reaching towards them and they are processing, and again you are supposed to do the action. If you are voluntarily do it, so the central nervous system is there and the skeletal muscle is being affected. And but if there is some motor activity in your body but involuntarily, so can you tell me which system is going to address them? Yeah, autonomic nervous system. Motor activity, how? The smooth musculature. Either in your heart, the heart beats, we never know. The GIT, smooth muscles. The respiratory muscles. All the motor activities being done along with the glandular secretion, right? But we are not aware of them. While sleeping, while doing any activity, all these are running. Motor activities are under control of autonomic nervous system. You must have the concept before, but the cerebral cortex is appreciating all these in a sense that we can do the voluntary action, right? And you always recall the thing you have already learned. The CNS is there, and you have got the sympathetic chain and parasympathetic system, that how they are gonna be communicating. They are functioning autonomously, but they are linked to your CNS in to an extent that if we say, if there is a condition, you are in the exam, you have got fear, you have got fight, flight, fright. But if you calm yourself, if somebody tells you it's just an exam, you will pass through it, there is nothing to worry. To a little bit extent, you overcome your fear. Your heartbeats try to settle down, right? So the things are that automatic or autonomous activities are being checked through your CNS. So that is the concept. So again, back towards your cerebral cortex, it's gray matter. Again, this picture is showing you the supporting cell. Which can you help me name? Astrocytes, okay, yeah, oligodendrocytes, microglia, right? So you can mention them all. Okay, astrocytes could be of two types, the protoplasmic and fibrous. So protoplasmic is here. In the gray matter, and the fibrous, as the word shows fibers, so would be in the white matter. So the details you can learn. So, okay. Again, you can see the what is the function of this main oligodendro? Yeah, in the, yeah, because there is another cell called as Schwann cell in the peripheral nervous system. So that happens. Okay, this picture is showing you again a central section, but tells us about that which structure is older in its version and which is new. So let's explain that we have got 6 layers of your cerebral cortex, right? Okay, but the older type of your cortex, which is called as allocortex, and it is having just not more than 5 layers. Could be 3, could be 4, 5, right? Or 1, 2, 3, 4, 5, and is now just right present in your olfactory area and the hippocampus. Only these two places will address this allocortex. This is simpler and primitive in its function. That can be asked. Okay. So the latest is what? Having the 6th layer is the neocortex. So easy to remember. NEO, you can abbreviate is with the new. NEW. Just to make remember that it is the latest one, and structurally and functionally having 6 layers, that allocortex is older, right? And if you remember the word HO, which I mentioned, olfactory, and the hippocampus, so you can make the mnemonic the house officer. The first job offered to you when you will inshallah pass through this MBBS, that you will work as house officer. The first simpler form is HO. Now you will remember that. that where lies the allocortex. There is another word you can read, the mesocortex, that is showing the middle in between the features of the allocortex and the neocortex. M is again for middle. That helps you. Okay. That we'll discuss later on. Okay, this is the slide of cerebrum. In actual, we will see later on with the help of this. That is just to show you the width of the gray matter. This is just 3 to 4, you would say, mm of it. This is the picture showing you the neuron, how the dendrites are there, and how the axons are there. But you observe the word the Schwann cell. So that could be the cell of this peripheral nervous system. Okay, now, as we mentioned, that the cortical neurons, if somebody asks you how you have to categorize the pyramidal cells, which are small, medium, and large, right? And the non-pyramidals, the horizontal, right? The stellate or ganglionic, and you can make one of it, if I put a finger like this, that could be a Martinotti, this cell, and this is the fusiform. Okay, so horizontal cell, you can see that is lying very superficially. So let's just take the name of these 6 layers. The layer number 1, which is superficial most, is the molecular layer. The layer second is external granular. Then the layer third is external still but pyramidal. Then you get the 4th layer. Now comes the internal word but granular. Now 5th is again internal but pyramidal. The 6th one is multiform. Right? We will see other pictures too. Here, what is the thing that is important? Thus, this superficial, the first, the molecular layer, is having all the processes in its parts. So much denser with the cell processes. But the cell bodies could be, or few cell bodies or cells you can observe could be the horizontal cell or maybe few neuroglia. इसका मतलब है ये layer जो है that is the synaptic zone जहाँ पे आपको processes दिखाई दे रहे हैं. Okay, why it is called molecular? That if these are few processes you can see longitudinally, but if you cut, how they are appearing as a dot or molecules or like sand. So this punctate or dotted or molecular appearance is giving the name to the first layer as molecular layer. So if somebody asks what it carries the transversely cut processes being shown as a molecule. And few cells or with the cell body could be a part of it. Okay. If you observe, there is another variety, the stellate cell. This stellate cell have got a cell body which is starry in its appearance because it is giving so many nearby processes making the cell shape as a star. So either ganglion or stellate cells, right? Having short dendrites and even the axon, and you can see that axon they are making connection with the other pyramidal cells you can mention. And these stellate and ganglion cell receives extrinsic or other input from your thalamus, right? And after that, we can discuss the cell of Martinotti. This cell have got a cuboidal cell body, so many processes, and its axon ascends upwards, right, to make connection in the most superficial layers, right? Okay. Then we have left with this fusiform. This fusiform is placed right angle to that of the surface. Okay. What happens? It gives dendrites from both sides, but from the middle of it, middle of the center, it will be giving the axon. Okay, so the things in first glance that the horizontal cells are lying horizontally, right, in the superficial layer. Fusiform, they are occupying the middle region, you would say, or even the deeper one. They are right angle to it. Other cells are normally placed. Okay, let's find some other pictures too. These are the type of neurons. If you can learn that how they are appearing and what are their names, you have learned in first year as well. Again, neuron making the synaptic connection with another neuron. This is how they are transferring their information to the other. Like if you've got hands, your body is a perikaryon or cell body. You are making connection with the other colleague, with the other colleague. This is how the chain is maintained and you are getting signals and being transferred. And finally, you all decide what to do. Okay, right? Again showing you various varieties which you have learned, the multipolar, bipolar, uni or pseudo, and other variety that either they are having connection with the exonic neuron sort of. There are various types of classification you have learned in your first year. That was just a picture to make you clear. Okay, that is just to tell you about these Roman numbers are telling you the sixth layer of cerebral cortex, right? Let's find some picture that would show the names. That is again the labeling of the neuron. Yeah, this is if we say your cerebral cortex gray matter. The covering we'll discuss later on, but let me add here, okay. Your brain is being covered with the meninges, you must know. The most outer, toughest is the dura mater, tough mother, durable, having two, again layer, endosteal and the meningeal, isn't it? Okay, next to it you get the arachnoid matter. Is this arachnoid is well vascular or avascular? Avascular, very good. Next to it, you get the pia mater. The pia mater is adherent to your brain tissue, right? That is again well vascular. So, okay, here you see that this is the layer 1, then you get the layer 2, then you get the layer 3, the 4, the 5, and then we enter into the 6th one, and then the white matter. So, something here you can see this transversely or horizontally running fibers. We will discuss that in which layer they are present. Just keep in mind that these are the bands of Billarger, these horizontal running processes. Let's see in some other picture again a beautiful color diagram to show you the layers. Okay, this is being lit. If you observe them and they are getting connection, that's a sketch we will do. Again the varieties. Okay, this is to make you remind the names. First, the molecular layer. You are now well aware that they have got processes being cut transversely, okay, and giving the molecular appearance. Then, having the horizontal cell. Okay, we enter into the next layer, which is external but granular. So, they are showing densely packed granules or stellate cells, which we already saw. But what else is there? They've got pyramidal cells, but of smaller variety, right? It means if we start from superficial to deep, the smaller size pyramidal cells will be towards superficial layer, right? Okay, now move towards external pyramidal. It will be addressing the medium size pyramidal cells, right? Okay. After that, it may be having along with it some granule cells, maybe having some Martinotti, other type of variety, fusiform could be there. Then the neuroglia or supporting cell could be apart and segregated and spread along these layers. Then you enter into the internal but granular. Means again granular cells take over. They are maximum in amount, densely packed in addition to the other cells. Like as I have mentioned, the neuroglia could be there, the Martinotti, etc., etc. Then after in this, stay here. This fourth layer will be addressing some transversely running fibers. They are called as band of Billarger, which I saw you in the previous picture. That is called as outer band of Billarger, which is the fourth layer. Then comes towards this fifth layer, which is internal pyramidal, addressing the larger, you can call it as bed cell, giant type of pyramidal cells. No doubt only this cell is not there, there are other cells as well. And here again, their processes will be making transfer horizontal running axons, the inner band of Billarger. It means the internal layer, this one superficial is making outer band of Billarger, and this will be showing the inner band of Billarger. Billarger is word maybe of the scientist or discoverer who discovered these things, okay? Then we enter into the last but not the least is the multiform. Simple in its explanation that it will be showing almost all variety of cell. Martinotti, fusiform, horizontal, pyramidal, right? So, again, if I help you to make initial, the first layer was the molecular. Okay. The second was external but granular. The third was still external but pyramidal. Fourth layer was now internal but granular. This one we make. Now again internal but pyramidal. This fifth one. Okay. Now coming towards the sixth or last, which is again with the starting with the letter M, multiform. So isn't it easy that starting with the M, ending with the M, two with the E and two with the I. You can make M, E, I, and then M. Any mnemonic you can make. Just to make it recall, because when you are laden with so many longer topics, so it becomes difficult to remember the name. But it's frequently being asked in Viva, or a written note can come that discuss the 6 layers and the cells residing in it. How would you remember them? That if you are all the neurons, I can label them first row as a pyramidal, smaller, then the next layer, the medium. The third could be the larger, still pyramidal. And I can address again the layer with the various cells, or we can name the non-pyramidal cells in which horizontal, the Martinotti, the fusiform, and what else is there? The granular stellate. So this is how. And I have arranged you into 6 layers. You get the concept that you are the workers, you are the main team, you can sense the thing, you have to make the orders. You are the governing body, right? Now you are arranged in different layers to have a seat, to do the function accordingly, right? And you are now the commanders, you are the chiefs to do all the things that are received by you, and then you take action. Is it clear? And you all are sitting in this area which is called as gray matter. Is it clear about the concept? Okay. Any other picture that helps you? Okay, granule cell, individually you can see a stellate shaped body, small dendrites and an axon. That is a horizontal cell line transversely having so many dendrites that ramify near the cell body, and again the axon moving towards the other side. This is the fusiform lying just right angle to the surface, have got the axon from the middle of its cell body, and the other two sides will be addressing the dendrites. Now the last cell, this is Martinotti with the cuboidal, we would say, cell shape, having so many dendrites, but an axon longer, that has to ascend, right, and take synapse on the superficial layers. Again, a histological picture to make you clear that how the neurons appear and how their processes you can observe. Again, a black and white diagram, but helps you a lot. That first layer is showing the horizontal cell, and rest of them are just synaptic zone, the molecular layer. Then you observe the external granular, in which granular stellate cells are residing. Then you enter into the third layer, which is external, which will be showing the pyramidal cells. Of medium size, the Martinotti cells are also resting. Then you enter into the fourth layer, which is now internal, and will be addressing this granular, the stellate or granule cells. Then you enter into the internal pyramidal, larger pyramidal cells, and other cells do take part, or they lie over there. And then the final, the multiform, the sixth layer, it will be showing if you draw some of the cells here. And now you can observe that afferent through white matter are reaching towards them, and they are deciding to do what? And the efferent is coming down. This is the concept how your brain works. Okay. Again, a schematic picture. Again, a colored diagram to show you the details. Okay. I think, let me show you the slide. But before going, just break here, I have to show you the other picture as well. Few other. Let me find the folder. Something. Have you heard about the word blood-brain barrier?
Yes, ma'am.
So can you help me to tell that what structures are going to form that blood-brain barrier?
Endothelium.
Yeah, endothelium, very good,
Basal,
of the blood vessels.
bas,
And
basal lamina,
yeah, it's basal layer. What else?
and
And among the supporting cell, the astrocytes, it's perivascular and processes you can see are engulfing these blood capillaries, the perivascular feet. Peri means around, the vessels, simpler term, right? And you can see the protoplasmic astrocyte, they are over it. And this is how, no doubt, the endothelial lining of this blood capillary, they are tightly or bounded with the occluding junction, right? But it is also making sure that no structure without permission should pass. So if a note comes to explain that what blood-brain barrier is, so you have to discuss. No doubt, few things can pass easily like water, the carbon dioxide, things like that, and we need glucose for our neurons and other amino acids. All these could be either simpler, they are supposed to pass, or they actively transported, or some of the substance they need permission or they require the receptor mediated transport that you will know in your biochemistry. So keep in mind that the things that are reaching, but the only thing that we are neurons, they consume as a food is the glucose, right? So, can you correlate the thing, the diabetes mellitus? Whenever there is hypoglycemia, whenever a patient comes towards a clinic, or you have seen a patient which is suddenly fainted, so what one or two steps you have to do? First, you have to elevate the foot end to make sure that to make blood flow towards the brain, one thing. But the other, you should be kept in mind that if it is hypoglycemia, you have to correct the glucose level. So you may put some dissolved glucose sublingually if you don't have any other injectable things. Otherwise, 25% dextrose will survive the patient, and no time he will or she will recover. So the hypoglycemia, if it prolongs, it is a deadly condition, right? So it's very important to address all of a sudden the unconscious patient, because our neurons are, this is the supporting cell, but our neurons, they just depend upon the glucose for their food, in a sense, especially in those diabetic patients, they need. Okay. So you are looking into another picture or diagram or topic, which is your spinal cord, right? Okay, few minutes because it's related. So if we take the transverse section, the reverse happens. What reverse? The central area is the gray matter and the outer has become the white matter. It is just opposite to that of your cerebral cortex. Okay. Now, what you see in the center, you have got a central canal, right? And what else? If you see that it's oval structure, and anteriorly it has got an anterior median fissure that is present, but posteriorly it is just in a form of a posterior median sulcus, and on its margins, what you see, the another posterolateral sulci are there. Here also dividing this white matter into various columns. These are called as dorsal white column because they are present dorsally. And they are called as anterior white column, and this is the lateral white column. So in combo or combination, it is called as anterolateral white column. And this is again anterolateral. Bilaterally we are studying, right? And these both are dorsal white columns. Now come towards this gray matter, which is showing the posterior horn, the anterior horn being connected in between through two commissures. The anterior gray matter, which is present anterior to the central canal, is anterior gray commissure, and posterior to it is the posterior gray commissure. Okay. Again, we should know that they carry what? They are having the neurons in it because it's a gray matter, well laden with the neurons, motor neurons, and some supporting cells could be a part. And of course, the initial segment of unmyelinated myelinated axons could be there, but predominantly we would say that neurons, they reside here. And what carries this white matter? They have got collection of the nerve fibers which are axons. But no doubt you can have some oligodendrocytes intervening and they could be giving the myelination around it. So that is the general transfer section. And another important thing, if you are in the thoracic or upper lumbar region, you get another horn called as lateral gray horn. Very good, right? What it is meant for? If this dorsal horn carries the sensory information or some sensory processes neurons, this was posterior and this is anterior having motor neurons. So what about this lateral horn, if we can demarcate? What it carries? Yeah,
Sympathetic.
it carries the preganglionic fibers of the sympathetic system. If you can recall, the thoracolumbar outflow of the sympathetic system. So the lateral horn is just in your thoracic region and upper lumbar region, right? Not in the cervical, not in the sacral region. That should be a concept. So a few other related pictures if we get, just discuss one by one. Again, a transfer section to make you recall the spinal nerve, the dorsal root, the dorsal ganglion, the ventral root, making a spinal nerve. Again, a transverse section with all these features. You can see the fissure is addressing beautifully the blood vessels, right? The artery, the veins are there. And if we talk about the coverings, the dura mater is outermost, then again same, the arachnoid just as that of your cerebral cortex, and the pia mater. Pia mater is tightly adherent to it, right? So that was about the meninges. Again, with the label diagram. Yeah, this is a picture to show you if there are neurons, what they carry, the nucleus, the nucleolus, and the cytoplasm is addressing the Nissen's body. What are they? Yeah, very good. Rough endoplasmic reticulum start with the ribosomes. So easy if somebody asks you or define this term. Okay. Yeah, this is just to show you that how these neurons and the supporting cells are present and being spread all around, right? You get the covering first, the pia mater is there. Then you initially observe the cell called as the astrocyte. Can you see the end? The perivascular feet are clung to this endothelial lining. And if this is the neuron, and you can see the oligodendrocytes, they are being wrapped up to give the myelination, right? And among these, you also observe the microglial cell. So this is how these cells are lying. They are making connections and they are doing function. ये देखिए myelination कर रहा है, ये अपना कोशिश कर रहा है to make blood brain barrier बनाने का, microglia है to deal with the or phagocytose various material which is not required later on. Yeah, this is the picture to show you that if this is the gray matter, so you observe various neurons which are multipolar and their initial segment are the processes, right? And what else? You can see the dotted, these are the neuroglia intervening. All these, so gray matter is filled with the neurons, right? And few initial segments which are called as processes, the dendrites or axons could be there because they have to make connection or they have to come out axons in the white matter. So keep in mind, सिर्फ ये नहीं हो सकता कि आपको cell body है तो उसके connection ही नहीं है. अगर उसके processes नहीं होंगे तो how they will communicate? So a some part could be there, लेकिन predominantly ये बैठे हुए. सही. इसी तरह अब ये एक्सॉन्स बाहर आई हैं व्हाइट मैटर में, आपको ये सारे ट्रांसफर सेक्शन मिल रहे हैं ऑफ दीज एक्सॉन्स बीइंग रैप्ड अप विद द माइलिन शीथ. इंटरवीनिंग कहीं पे आपको ओलिगोडेंड्रोसाइट्स भी दिखाई देंगे. तो लेबलिंग के लिए, ड्राइंग के लिए अगर आ जाए तो यू हैव टू सेग्रीगेट हाउ द ग्रे मैटर एंड हाउ द व्हाइट मैटर इज. Okay, again another schematic diagram to show you the things. Blood-brain barrier. The picture of the oligodendrocyte. Let's skip these because I have learned, you have already learned them. Okay, the white matter, what it carries? Myelinated axons, but some unmyelinated could be a part. The neuroglial cells among these oligodendrocytes. So if a note comes what white matter is, you have to mention all these. And if a note comes what gray matter carries, you have to tell that. Neuronal cell bodies The dendrites could be an initial axonals and some neuroglial cells, right? So this is how you categorize and write a note if it comes. Okay, another important, interesting thing, that we've got a spinal cord, but its thickness varies. If you are in the cervical region, the maximum girth is there, right? And if you then enter the thoracic region, a little bit, but a narrower, a little bit portion. Then you enter into the lumbar region. It is the size is being shrunk, right? And when you enter the sacral, the white matter again is. Can you give any good reason for this? The fibers,
area of
ascending fibers are keep on adding from below upwards, and descending fibers are jumbling, coming downwards. So the area that is receiving maximum girth is due to these bundle of axons. This is one of the reasons. Okay. So we should know that motor supply, what they do, and we another observe a thing that let me find another picture to make it clear that what these enlargements are. This is the slide of it, again to show you the various. Okay, this is the spinal cord. You can see it has got definitely the cervical region, thoracic, lumbar and sacral. So if you can observe that the lower end of medulla oblongata, later on it becomes the spinal cord, and up to the L1-2 level, vertebral level, it ends. Your spinal cord ends, and then it the tapering end is called as conus medullaris. Then it continues as a phylum terminale, having phylum terminale externum and internum. Total 20 centimeter, but 15 is externum, internum is 5. Then you see that fibers, these spinal nerves initially take the horizontal course. Later on they are doing the oblique course, because spinal cord is going to end at some upper region, but you have got nerve supply in your lower limb as well, and your pelvic region. So they are now taking the vertical course, hence form the tail-like appearance of horse tail called as cauda equina, right? And if somebody asks you that at which level your this spinal cord ends,
L2.
you see if your this you can mention in between L1 and L2, but if your elbow, if you place it a little bit above on your body, your spinal cord ends, and it could be at the transpyloric plane. And you know what pylorus is, terminal part of your stomach. So this is how the landmarks helps you कि इंजरी किस लेवल पे हुई है, तो स्पाइनल कॉर्ड का कौन सा हिस्सा जाएगा, उससे नीचे है तो क्या होगा, और बहुत नीचे इंजरी वर्टिब्रल कॉलम है तो मे बी स्पाइनल कॉर्ड सेव हो गई हो. This is the logic behind it to learn. Okay, again a label diagram. I'm just dealing with those that are concerned. Okay. Now you see into the picture, just to make you that the pia mater, the innermost, giving on its both sides the transversely running ligaments. They are called as dentate ligament. 21 in numbers they have mentioned. That picture is just to make you how your body is receiving various sensations. They are being ascending through this spinal cord, then the upper region of the brain, the medulla, the pons, the midbrain, and finally reaching through the thalamus, because it's a relay station, and the sensory area is receiving them. This is a key map to show you how your body gets the info, and the process is there and you act accordingly. Again, this should be telling you that receptors, so carrying first order neuron, you entered the spinal cord. Here they have got relay nuclei. This is a schematic representation. Then when the other neuron is there will be second order. When you enter into the thalamus, the neuron is the third order. Finally, in the cerebral cortex, finally towards the destination, that could be fourth order. So this is how first order, second order, either sensory are moving or motor information is coming downwards. Again, this is in repeat. That was to show you the ascent and descent of the fibers, the white and the gray matter. Okay, just I'm looking for the two. Okay, I can just otherwise tell you that our spinal cord in the cervical region, at the level of that C3 up to T2, you have got cervical enlargement, right? Because it has to give rise to nerves of your limbs.
upper limb.
Then you enter into the lumbar region. It again will be addressing the lumbar swelling or enlargement at the level of, you would say, the L2 and then sacrum 3. That is the, you would say, the spinal segment level, right? So you can make the number in the cervical region 32, and below you can, if L1 is added, you can make 13 in a sense. So the cervical enlargement in the spinal cord and the lumbar enlargement, because the upper limb nerves are through cervical enlargement and the lumbar enlargement is addressing the nerves for your lower limb. So that was the picture. If I can get anywhere, that was just to show you the central nervous system and the peripheral. Okay, that picture was just to show you that the end of spinal cord is conus medullaris. Then it will be running as a phylum terminale. It has got the externum and internal portion, and okay, another important thing, that the end of the subdural in the subarachnoid space is S2, right? So you should be knowing that the spaces lie where. Why spaces are important? Because below the L1-2 level, whenever we have to do the LP lumbar puncture to get the CSF, we preferably select a level L3, L4. We put a patient in prone, semi-prone position in a full flexed, so that the vertebrae, they should be in full extended form, spinal cord that moves upwards, right? So you get a good plantar space and you take LP either for diagnosis, either for therapeutic, either to give any medication or to take sample. So these are the related things. Unluckily, I'm not getting them. Let me find the... Okay, that was just to show you that you have got cervical, thoracic, lumbar, and sacral segments of your spinal cord, and of course vertebrae are there. So it's very important to learn that at which vertebral level, which spinal segment rests. You will learn in your brain region, and definitely it will be there to work, that to show. I think let's finish because no other related diagram is... Okay. Okay. Now let's see the histological pictures. Okay. So in this staining, which is silver staining, on its very outer side, we get, if this is the arachnoid matter, then we get the pia matter, right? And this is the, these are the gyri, right? And this is one of the sulci or sulcus, if plural or singular word is sulcus, and the plural are the sulci, right? And we enter through this, that could be first is the molecular layer, then we enter into the external granular, right? Internal pyramidal, then external pyramidal, then word internal granular, internal pyramidal, and the multiform. So there is, you see, there is no such line. Allah Ta'ala ने ऐसे फुटे से लाइनें नहीं बना के भेजी. The cell series और उनकी variety बदल जाती है, तो we know कि in which layer we are. So from superficial to deep, we mention the 6th layer. So keep on rotating till we reach in the core. That is showing the fibers, the white matter. You see, from this side, all the gray matter. Let me focus it a little bit more. From all this, the similar layers are approaching, so full of neurons, and these fibrous or axonal portion will be addressing the white matter in the center. So usually for your OSPE, we focus on such place that helps you the look of the gyri and the sulci, right? And there are coverings too. And one of the good thing that it is in the silver stain, so that helps you to identify the cerebral cortex. So we can rotate it from anywhere. So various pictures. So in first glance, this is the gray matter. In the very center, we observe the white matter. So now let's change the slide. Okay. This is the slide of what? Spinal cord. Very good. You have now learned enough. So this is the anterior portion because it shows a wider fissure, the anterior median fissure, right? This is the central canal. Let me focus it a little bit more. Okay. Again in a silver stain. This one is the dorsal or posterior median sulcus. These are the a little bit apostolatal sulcus. So now about the gray matter. This is the ventral horn. This is the dorsal horn, and if it is the cervical or thoracic region, we can have here the lateral horn. So this is the anterior gray commission, the posterior gray commission. These are all this gray matter is filled with the neuron varieties we have discussed a lot, and these are whole the axons in the white matter, myelinated, so they could be having the oligodendrocytes along with it. So that was all about for today. Thank you very much.